Hello and welcome back to your nutrition class. Today we want to learn about our nutritional needs. First, let's focus on our objectives. How do we determine nutrients needs? What is dietary reference intake? It's referred to DRI. And food labels usually. Estimated average requirement. Recommended dietary allowance. We see that quite a bit in the food label. What would be adequate intake of nutrient? What is the tolerable upper intake level? As well as what's the estimated energy requirement at the daily basis. Acceptable macronutrient distribution range. Remember, we studied macronutrients, which include food groups, carbohydrate, protein, and lipids. How much do we need them per day, and why? An interpretation of nutrition research, the data that we read in journals, scientific journals. How can we analyze them and understand them? As well as evaluating nutrition-related claims. The credible claim compared to claims that are not made by scientists. And what are the functional foods? Functional foods such as foods that are not regulated by FDA, Federal Food and Drug Administration, but we need them because in 21st century, we have lost many of the vitamins that our body may have been producing in the past or other things. We get to that later on, not to worry. So let's look at this slide. How do we determine nutrients needs? DRI, which stands for Dietary Reference Intakes, identifies the amount of nutrients that we need to prevent deficiency diseases. We talked about a few of those. Lack of vitamin C, lack of vitamin B12. Furthermore, the amount of a nutrient that may reduce the risk of chronic diseases, such as heart disease, diabetes, high blood pressure, as well as upper level of safety for nutrient intake. Let's review them and study them together. Move on to the next slide. Dietary reference intakes consist of 4 values usually. Number 1 would be the estimated average requirement. Then would be recommended dietary allowance, adequate intake, and tolerable upper intake level. We'll review those. But we want to a little bit focus first on average requirement as well as food safety. Let's review this graph. There are 2 bar graphs. On the left side, the bar graph shows inaccurate view, which usually general public considers that would be what's considered in our food that we consume. safe or dangerous. But the accurate way to look at the food that we really consume involves safety, marginal safety, danger or deficiency, and as you move up, the bar graph on the right side would be danger due to toxicity. Let's review them together. Take a few seconds first to look at the bar graph on your right-hand side and compare it with the inaccurate bar graph on the left side. Focus on the safety range. Most common people believe food is pretty much 80% safe, but in reality, it's a very narrow range here in an accurate graph for safety. Type of food that we consume that will not jeopardize our safety. Let's move up first from safety. There is a very marginal level that we can still tolerate it, but it's not very safe. Above that, food could become toxic. So what can contribute to toxicity? Additives, preservatives that are added to the food. For example, when you consume cereal, when you look at the cereal box, has a list of vitamins, minerals, and food groups that they can't possibly be in any type of given food at the time. So they're all added, and they actually could contribute to toxicity of the food. It could end up to be more than you and I need. And if we don't take sufficient nutrients in the food, let's move down the graph. There's a marginal level that we call it estimated average requirement, below the estimated average requirement. And then would be if you don't consume sufficient vitamins or minerals, then it will be danger of deficiency. So let's review what we said. There's a safety range for the food, very narrow. Above that would be tolerable upper intake level, what your body can tolerate but is not good for you. Exceeding that level will become toxic. There is a recommended daily diet, which should fall into the safety range. Furthermore, is the estimated average requirement. If we don't get sufficient food and nutrients, then we may suffer from deficiencies. Okay. Take a few seconds, put them together. View your slide again. We move on to the next one. Estimated average requirement would be the average daily intake level of a nutrient. That's based on meeting the need of half of the healthy people in a particular life stage and gender. That means would be elderly, teenagers, children, male or female. An EAR is used to determine the recommended dietary allowance, RDA, of a nutrient. Let's move on further. What is recommended dietary allowance? Would be the average daily intake level that's required to meet the need of pretty much between 97 to 98% of healthy people in a particular life stage as well as gender group. So the dietary allowance is much more credible and accurate because it involves higher percentage of studies that they have done among people to produce those data. What's adequate intake? Well, adequate intake would be recommended average daily intake level for nutrients. And that would be based on observations and estimates from experiments that they have done. So used when RDA is not yet established for a certain type of mineral or vitamin. So is not a required dietary value that we need to have it per day. It's not established by FDA, Federal Food and Drug Administration, but we need it, such as calcium, vitamin D, which is required to absorb calcium, as well as vitamin C is required to absorb calcium from your blood circulation, vitamin K, and fluoride. So we need to take a sufficient amount to stay healthy. What's the tolerable upper intake level? UL stand for that. Would be the highest average daily intake level that is not likely to have adverse effect on the health of most people. That means you can't exceed that level. You can tolerate it, but you cannot exceed that level. And once again, this is very individual, related to age. Obviously, a child cannot have the same tolerable upper intake level as compared to an adult or elderly. So consumption of a nutrient at levels above UL, that means tolerable level, is really no longer considered safe. Let me give you an example. It's vitamin A. Vitamin A actually is very beneficial. It improves our vision and is involved in many other chemical aspect of metabolism. But once we take more than tolerable upper intake, it can become quite toxic. Actually, it could cause birth defect, abnormalities in unborn fetuses. Let's review now estimated energy requirements. We refer to it as EER. How much energy do we require? And then later on, we'll discuss how much energy do we get from a given food group that we consume. Average dietary energy intake to maintain energy balance, it depends. It depends on your age and your gender, your weight, height, as well as the level of physical activity is extremely important. So, in summary, we want to say our energy requirement is basically very individual. Must fit our lifestyle. Let's move on to the next slide. Now we want to review acceptable macronutrient distribution range. The range of energy intake from macronutrients that are, what are those? Carbohydrate, lipid or fat, protein, associated with the reduced risk of chronic diseases. Those are must-have, that the level is required by FDA. Furthermore, the range of macronutrients intake that would provide adequate level of essential nutrients. So in addition to energy, they supply quite a bit of nutrients. How much carbohydrate would you need to consume per day to be healthy? How much fat or protein? Let's review those. Let's review them in this slide. The acceptable macronutrient distribution range. About 45 to 65% of your daily diet should include carbohydrate. But it's important to distinguish between complex carbohydrate and simple carbohydrate. The best way to consume carbohydrate would be complex. We will study those in other lectures later. The fat intake should not exceed 20 to 35% of your diet. You actually don't need as much protein as people claim that you should consume because you're physically active, or protein can really sustain growth. That's absolutely true. Protein is extremely important, but should not really exceed 10 to 35% your daily diet. Excess intake of protein will have negative consequences as well. But please note, this is a reminder that proper healthy food will not be giving you excess nutrients, macronutrients, or minerals or vitamins. Supplements do. Protein shakes, protein powders, they could be excessive. Now, let's review together interpretation of research result, nutrition research. How do we conduct research? What's a reliable way to research nutrition and then produce credible data? Well, research involves applying the scientific methods. We want to start out by first observation, then hypothesis, conducting experiment, collecting data or producing data, and coming up with a conclusion. And that's done by scientists. That's how scientists do that. Scientists usually have curious mind. Suppose that they are investigating food in grocery store. So they are viewing first, that would be observation. Based on observation, they're looking at fruits and vegetables such as apple, let's say, as an example, and comparing it with bacon. So we're literally looking at them superficially. We can't tell about their content. So they come up with a hypothesis. Let's say my hypothesis would be bacon tastes better and makes me full very quickly, possibly gives me more energy. So I think bacon is better for me. But as a scientist, that would not be sufficient. I cannot really produce data that is publishable. So I need to design, experiment, and repeat it many, many times. Extract the nutrients in the apple, and the content in bacon and compare them together. The fat content, the protein content, and carbohydrate content, as well as the additives, chemicals that are added to it. And then I produce series of data. So I need to analyze those data. Those would be my result. And based on analyzing data, I realize and conclude that my hypothesis, initial hypothesis, was incorrect. So would be formation of a conclusion. And based on my hypothesis, I need to do the experiment over again. And propose an alternative hypothesis. So that means my next experiment will focus on fruit and vegetable. Fruit and vegetable would be more beneficial for our health, and conduct further experiments. So in conclusion, development of a theory-based or repeated experiments lead to scientific data that we have access to. So in your next slide, we see a diagram that summarizes what we said. We just run very quickly through it. It's not quite accurate, but what we did discuss was accurate. Start out with observation, hypothesis. We come up with an educated guess, might be right at the end or incorrect. And then conduct series of experiments. And then here I would say after experiment, we come up with results, not observation as much. We come up with scientific data and results as a result of our experiment. In our case, data on the right side did not support my hypothesis, so I need to change my hypothesis and repeat the experiment. But there will be cases that data do support the hypothesis, and in this case, we come up, once we have an acceptable hypothesis, come up with a theory that we can publish it in scientific journals or publish it in the textbooks so that you can study it. Now, what are the different type of research studies out there? How do they study in the laboratory? Many ways. Usually, scientists do laboratory studies. One way would be animal versus human studies, observational studies, case control studies, or clinical trials. Well, why do they use animals and not human? Well, eventually they have to use them. Suppose they are studying a medication, a drug that's supposed to cure heart disease, or studying new antibiotics. They can't use human initially as a subject for ethical concern, as well as animals are not likely to hold us liable and sue us. But then once we do all the animal studies, our results may not apply to human. So ultimately, once the drug is approved partially, not completely, we go through a trial process and test our drugs or antibiotic form of drugs on human. How do we do that? Well, stay tuned. We'll talk about that. And then there are observational studies, much easier. In observational studies, can only indicate relationship between factors, like we did with apple and bacon. But once we took it to the laboratory, then they became real scientific studies. And then there are case control studies. What are those? And clinical trials. Let's review them. In clinical trials, an intervention's effect on a certain disease or health condition is tested using two groups usually. An experimental group and a control group. Remember, we said we are testing possibly a new antibiotic or a drug, a medication. So we break people into two groups, group A or B possibly. Let's say group A will be our experimental group, and we will use group B as control. Let's move down here and then we explain it further. And then we have randomized trials, as well as single or double-blind experiments. Double-blind experiments involves two group A and B that they completely have no knowledge of which group is experimental group or which group is being used as control. So in a control group, they don't use that given new antibiotic, for example, on patients. They use placebo. What's the placebo? An imitation treatment that has no effect whatsoever. It will be given to the control group. In the so-called placebo control group would be double-blind, randomized clinical trial. And then to the experimental group, they will administer antibiotics. Now, it's very important that we have a large number of subjects. There have to be thousands of people involved in this test. So if we see the experimental group does substantially well after being treated with this new form of antibiotics, so we can conclude that that medication or that drug is helpful, treats infectious diseases. If it doesn't, then it will not be approved by FDA and cannot be allowed to come to the market. Now, this is incredibly important. How do we evaluate nutrition-related claims? Are they credible, or they are merely claims? It is critical to ask the following questions. To determine scientific validity of the data that are presented to you, you want to ask, who is reporting the information? Where was this study done? Did they do it according to what we just discussed? Or it was just based on a survey? Furthermore, who conducted the research and who paid for the research? The tobacco company paid for the research to study safety of cigarette? then you have an answer for this question yourself, right? Here you go. Very important. Who sponsored that research project? Is there a conflict of interest? Let's look into it. Move on to the next slide. So the study that was done, is the report based on reputable research studies? Did they do it in an academic institution, a university clinic? Was there a control group and an experimental group studied in this process? Was the sample size large enough to rule out chance variations? We said you need to have thousands of subjects. To have your result to be conclusive. Was a placebo effectively administered to the control group? Was it a double-blind study? Or is the report simply based on testimonials? Do the claims seem too good to be true? If they are, they seem too good to be true, they probably are. In 21st century, most of you probably tap into the internet and research certain things. How reliable are information that you gather from website? Well, depends on who sponsors the website, if they have credentials. Whether the date of the website is recent, within the last 2-3 years. And there are few internet addresses that they seem to be credible. Those are internet addresses that they end with gov, so they are government tested, should be reliable. I do have my concerns. I do believe in EDU, academic institutions, and .org. So is it .gov, your governmental studies, .edu, your academic institutions, .org is generally non-profit organizations. Most of those are also academic institutions that they have hospitals and clinic and research institutions. They're considered, in general, reliable, but then yet again, we need to further study those on our own, compare them with many other data. Who can you trust? Well, when you want to really research nutrition and your health, trustworthy experts must be educated and have credentials. Examples: registered dietitians, licensed dietitians, nutritionists with credentials and experience, professionals with advanced degree in nutrition and health, as well as physicians or scientists with appropriate expertise in nutrition. Let's get a little bit more in detail, whom we can trust. Government agencies are usually supposed to be trustworthy, but you need to double check them. Example, the Center for Disease Control and Prevention, CDC, supports two large national surveys. One is the National Health, and the other one is Nutritional Examination Survey. They're pretty reliable. There are also behavioral risk factor surveillance system survey. They also do survey studies, as well as numerous institutes within the National Institutes of Health, NIH, that focus on a specific area of health. They actually are a very good source of information, and they're grant-based. So the area of the research they evaluate include cancer, heart disease, lung, blood disease, diabetes, as well as alternative medicine. So NIH actually provides grants to scientists to engage in those studies, and they have large number of scientists that they evaluate those studies, and the studies must be reproducible. That means a separate independent institution ought to be able to reproduce the very same data. Well, are those the only one that we can trust? Well, there are also some professional organizations that publish cutting-edge nutritional research and information. It would be very difficult to really Google information and then come up with reliable scientific data. Science should be facts-based. Few of those professional organizations are Academy of Nutrition and Dietetics, American Society for Nutrition, American College of Sport Medicine, North American Association for Study of Obesity, and there are a few more organizations that they are pretty reliable. Let's see what's considered as new frontiers. What are the new things that they have discovered? Really interesting. Nutrigenomic has to do with genes and nutrition, combination of nutrition and genes. Can your nutritional needs be regulated by genes or vice versa, or what you consume, can they have an effect on the gene? Let's review this slide together. Nutrigenomics studies the interaction among genes, the environment, and nutrition. Key theory is food and environmental factors can switch some genes on while turning off other genes. What are those genes? Can they be cancerous genes that we have inherited? Can they be turned on by the food we eat or turned off by the food we eat? Absolutely. There are actually a lot of data on that. It gets more interesting. They could help in reducing risk of diet-related disease, treating existing condition through diet, and making personalized nutrition possible. Let's study the next slide, which is actually quite interesting and support what we just studied. Among new frontiers, there is a project, we call it Human Microbiome. Let's see what it is. First, what's human genome? Human genome is a set of genes making up your DNA in the nucleus, or my DNA in the nucleus. We call it human genome. Human genome project was completed in early 90s. But human microbiome involves microorganisms, bacteria. It's a set of genes belonging to microorganisms that live in the human body. So those microorganisms are considered as normal flora, referred to as GI flora. They live in your gastrointestinal system, mostly intestinal system, large and small intestine. And they benefit you. How do they benefit you? First, they provide enzymes that they can break down fiber, which we consume in fruit and vegetable. In addition, those microorganisms produce vitamin K, which is involved in blood clotting, variety of vitamin Bs, especially vitamin B12, is required to make blood produce red blood cells. Is actually a lot more that these microorganisms do. They actually can turn on the good genes in your DNA and turn off the bad genes. So our health is affected by the way they interact with human cell and gene and your DNA. So is that safe to say that we need to maintain those bacteria? And allow them to stay in our gastrointestinal system? Absolutely. But unfortunately, we have lost those normal flora that are so beneficial in our gut. As a result, we depend on functional foods. So what are functional foods? They have ingredients with potential to provide health benefit beyond basic nutrition. They're not regulated by FDA, but they can help our digestion, our metabolism. You're probably very all familiar with probiotics. Probiotics are pills that we take, or powder, that contain those good microorganisms that we have lost to improve the intestinal microbial balance. How did we lose them? Well, the pesticides or Roundup that they are used on our crops kill the bacteria in the soil, which usually those bacteria enter our body through our fruits and vegetables that we consume, as well as they kill bacteria, continue killing bacteria in our intestine. In addition, we take too much antibiotic, and we are exposed to too much antibiotic, and excess use of antibiotic has destroyed our normal flora. So that we have to really take them in form of probiotics, which is very unfortunate. Is there hope? Yes, organic food. Organic food lacks pesticides and reduce the use of antibiotics. What are prebiotics? Prebiotics are non-digestible ingredients that stimulate the growth and or activity of helpful bacteria. Example would be fiber. What are phytochemicals? Phytochemicals are antioxidants, very beneficial. They're plant-based and may reduce the risk of disease and cancer. So we covered quite a bit. So I want to thank you for joining me during this lecture, and keep on working hard, and don't forget to have fun while you're studying nutrition. Keep up the good work. Bye-bye.